Hello everyone and welcome back for more biotechnology. So in this chapter we are finally going to start really discussing something that we have been mentioning throughout the semester, but we have not quite gotten to the point where we can actually start discussing it. Well, that day has finally arrived and we are going to start talking about the use of spectrophotometry. So let's go ahead and get into this. So in the previous chapter, we talked in depth about both the ELISA and the Western blot assays, which are good for identifying very particular molecules in a mixture of molecules. But what they rely on is that they rely on the idea that we have antibodies that detect those specific molecules. And here's the deal, not every molecule is going to have an antibody that can detect it. So ELISA and Western blotting is going to be a little bit limited in terms of what we can use them for. So what spectrophotometry is all about is that it takes advantage of the way in which certain molecules absorb with and emit particular wavelengths of light. So basically, we are working on the principle of how molecules interact with radiation. So different molecules will interact with light in different ways, and we can use this to identify certain molecules using this. So we've actually already seen this principle in action several times throughout the semester. So we've already seen how dyes like ethidium bromide, Coomassie, green fluorescent protein, all of these absorb UV light and they emit visible light, meaning that we can see them when we shine UV light on them. And you'll recall that ethidium bromide was there to make DNA visible to us. Green fluorescent protein is a protein that is already visible if you shine UV light on it. Coomassie makes proteins visible. So this principle of molecules interacting with light has already been basically right up our alley from the very beginning. And like I said, we've already experienced this in the Bradford assay, in which we used the dye Coomassie Brilliant Blue. And what we did in that particular lab is that we used a spectrophotometer to quantify the concentration of proteins in solution. So we were doing that all based on the way that those proteins and the dye molecules interact with light at a particular wavelength. So the two major types of spectrophotometry that are certainly the most common are called UV spectrophotometry and visible spectrophotometry. So as the name suggests, UV spectrophotometry will utilize ultraviolet radiation, and visible light will be for what visible spectrophotometry does. Alright, so let's talk about the principles behind how spectrophotometry works. So if a solution contains a solute or a molecule that absorbs light at a particular wavelength, then shining light of that particular wavelength through the sample should cause a certain fraction of that light that we're shining through to be absorbed, meaning that it does not make it to the other side. So if you look in the picture here, we've got a light source, which is indicated by the light bulb here, shining light through this sample, and you'll notice that a certain fraction of that light gets absorbed by molecules in the solution. Maybe it gets reflected off, but regardless, of the light that actually passes clean through the sample, it only represents a fraction of the light that came from the light source. So any good spectrophotometer will allow you to choose the wavelength of light that is passing through the sample. And we'll have more to say about wavelengths of light later on, if that is not a familiar concept to you. So this light at this particular wavelength, like I said, will either pass straight through the sample, meaning that it did not interact with any of the molecules. It could be absorbed by the molecule of interest, or it could be reflected off of other molecules like the solvent, usually water. So the percentage of light that actually passed through the sample is given as the percent transmittance. So if all of the light passes through the sample, none of it gets absorbed, none of it gets reflected, then your percent transmittance would obviously be 100. If none of the light passes through the sample and all of it gets absorbed, then your percent transmittance would obviously be 0. So we're not really going to use percent transmittance for everything. We usually like to deal in a different metric called absorbance, the degree to which light actually got absorbed. You can convert back and forth between percent transmittance and absorbance by using this equation here. 2 minus log base 10 of the percent transmittance. So what that means is that if your percent transmittance is 100, meaning none of the light got absorbed, then the value of log base 10 of 100 is 2. So the absorbance would equal 0 because 2 minus 2 equals 0. Whereas if all of the light got absorbed, then the value of the absorbance would be 2. So we'll talk later on about why the absorbance cannot be a number greater than 2. So the idea that we're going to be working with, and you understand this from having done the Bradford assay at this point, and we'll learn more and more about this as we go along, but the general idea of what you use spectrophotometry for is that the more concentrated a solution is for your solute of interest, the more light is going to get absorbed by the sample. So as those absorbance values increase, your molecule of interest, if you're shining the correct wavelength on it, the absorbance is going to go up and up as the concentration goes up and up. So practically speaking, samples are loaded into a plastic container called a cuvette. It's very small. It's maybe about half the size of your finger. So the cuvette is just a little plastic container that looks like a big rectangle. So generally speaking, it is situated such that the bottom of the cuvette occupies a 1 centimeter by 1 centimeter area. And why that 1 centimeter figure is important is something that we'll talk about later on when we talk about the Beer-Lambert law. But basically, just for practical purposes, your sample is going to be loaded into this little plastic container here, and that's called a cuvette. So if we're using a visible spectrophotometer, the spectrophotometer will generate white light, which is just a mixture of light of all the visible wavelengths, everything from 350 nanometers to 700 nanometers. And then a grating or prism will separate these wavelengths apart from each other, kind of like the cover of Pink Floyd's Dark Side of the Moon, if you're familiar with that sort of thing. So we split the light into all the separate wavelengths, and then depending on which wavelength we want to pass through the sample, the spectrophotometer will adjust a slit such that only light of a particular wavelength gets let through, meaning that all the wavelengths get blocked by this grating here except for the wavelength that we want, which is then what shines through and passes through the sample. So at this point, our light shines through the sample, and then what actually makes it through the sample and does not get absorbed or reflected will be picked up by a detector, which will get that information to the display up here and tell us exactly what the absorbance is. So visible spectrophotometers use what is called a tungsten lamp to produce just your regular old white visible light. So really not unlike a lot of the light bulbs and fluorescent lights that you have in your home or in your buildings. However, if you have a sample, if you have a molecule of interest that absorbs light at a wavelength less than 350 nanometers, you cannot do anything with a visible spectrophotometer. What you need is a ultraviolet spectrophotometer. Those wavelengths that are less than 350 are not in the visible wavelengths, they're in the UV range. So a UV spectrophotometer uses instead a deuterium lamp to be able to generate those wavelengths that are in the UV range, between 100 nanometers and 350 nanometers. So more on this later, but UV spectrophotometry will particularly be useful for looking at proteins and DNA. As we will find out later, proteins absorb light at a wavelength of 280 nanometers, that's in the UV range, and similarly DNA samples will absorb at 260 nanometers, also in the UV range. Alright, so we've been talking about wavelengths this, wavelengths that, so let's actually learn what a wavelength is. So light, if you've ever dabbled into physics at all, light behaves as both a particle and a wave. So we're not going to worry too much about the particle, we're more interested in light's behavior as a wave. So because light behaves as a wave, we can describe electromagnetic radiation being light in terms of its wavelength, which is just the distance in between consecutive waves. So some types of light have very short wavelengths and some of them have very long wavelengths. So that is going to be how we differentiate one type of radiation from another. by its wavelength. So importantly, the general idea is that the shorter the wavelength is, the more energy that particular type of radiation has. So if you look at the chart here, in which we start with the visible spectrum, which is in the 350 to 700 nanometer range, as we get even shorter and shorter wavelengths into the femtometers and so on, we're going to start talking about things like gamma rays, X-rays, UV rays, things that you probably know just from looking at those. Those are a little bit dangerous, so you have to be careful about UV radiation so your skin doesn't get burned. You have to be careful with X-rays when you go to the hospital. Gamma rays are very, very dangerous, and thankfully there's no way that we can really encounter them here on Earth. But because those types of radiation have such short wavelengths, they've got incredible energy associated with them, and if we encountered them, our flesh, our tissue, would be very highly damaged. Now, on the other hand, something that has a very, very long wavelength, like radio waves, because they have such very long wavelengths, they have practically no energy associated with them, and they are totally harmless to us. So even as I'm speaking and as you're sitting in front of your computer listening to this, our bodies are currently being bombarded with radio waves all the time, but it doesn't do us a lick of harm because that energy or that radiation has just absolutely such little energy associated with us, it can't hurt us at all. So we've already established that visible light, meaning the light that our eyes can actually see, falls within a pretty narrow range. Everything from 350, which is going to be the deep, deep, deep purple colors, and then up to 700, which is going to be the very dark red colors. Everything in between, you'll notice, is the blues, the greens, the yellows, the oranges, the reds, and so on. All right, so let's back up from biotechnology and just talk about something that is, I think, important for everyone to understand, regardless of whether they're in a biotechnology class or not. Why do certain things appear certain colors to us? So, to answer this sort of question, let's start by talking about that Bradford assay that we did recently. So that dye, Coomassie Brilliant Blue, turns a vibrant blue color when it is bound to protein. But what is it that actually makes it appear blue to us? Well, the answer to this is that when we see color, it is because the light that shines on what we're looking at absorbs certain wavelengths and reflects others. So the idea in the case of Coomassie, which we might be looking at in this picture here, you can see we have a bunch of blue molecules here. This dye absorbs all the wavelengths of visible light except the blue wavelengths. Those get reflected back at our eyes, and because our eyes are being stimulated with visible light that corresponds to blue color, that's what we see. Therefore, the solution appears blue to us. And of course, we can apply the same principles to things like why is the sky blue? Again, the molecules in the atmosphere must be absorbing all the wavelengths of visible light except for the blue colors, which are getting reflected back at us. It'll help us explain why plants are green, why carrots are orange, and things of that nature. Why are tomatoes red? It all has to do with what wavelengths are not getting absorbed and are instead getting reflected back at your eyes. So let's look at this in terms of the absorption spectra for particular molecules. So this is what's called an absorption spectrum. You can see on the x-axis we have pretty much the entire visible wavelengths here, everything from 400 to past 700, and on the y-axis you're looking at the absorbances at those particular wavelengths. So this is the absorption spectrum for Coomassie Brilliant Blue. This first one, labeled A, is Coomassie when it is not bound to protein, and the one B right here is Coomassie when it is bound to protein. So what you'll notice here, if we just focus on the Coomassie that is bound to protein, it does not really absorb any light until we get to about the 550-ish nanometer wavelength. So you can see we reach a maximum of around 595 nanometers. That may sound familiar to you because that was the wavelength we were using when we did the Bradford assay. And then it stops absorbing right when it gets past the visible light spectrum. So if we want to interpret this, we could say that Coomassie absorbs all the wavelengths except for those between 400 and 500. Those must be getting reflected back at us. Well, if we back up here a second and look at the visible light spectrum, what are we looking at between 400 and 500? We're looking at the blue colors. So, of course, that explains why Coomassie appears blue to us. It is not absorbing blue colors. It is instead reflecting them back at our eyes. Let's look at another example here. Let's talk about chlorophyll. Chlorophyll, as you I'm sure are aware, is the pigment that is present in plants and other photosynthetic organisms like algae and cyanobacteria that is not only responsible for photosynthesis but also responsible for those organisms appearing green to us. So if you look at the absorption spectra for the two different types of chlorophyll here, which are a little bit different, you'll notice that they do a pretty good job of absorbing light between 400 and 500, those are the blue colors, and also between 600 and 700, those are the orange and the red colors. However, light is not getting absorbed between 500 and 600. Well, guess what color that corresponds to between 500 and 600? Yep, it's going to be a lot of green in there. So for chlorophyll here, the fact that plants appear green to us, it must be because that chlorophyll is absorbing blue and red light, but not green. That green light gets reflected back at us, back to our eyes, and that is why they appear green to us. Okay, so let's talk some more specific biotech stuff. So we're kind of past the evergreen nature of this discussion, and now we're really just kind of focused in on biotech applications. So a given molecule, so if you decide you want to use spectrophotometry to look at a particular molecule, you pick your molecule. So let's say that your given molecule has a particular wavelength of light for which it absorbs the maximum amount of light. So every molecule is like this. Every light-absorbing molecule has some particular wavelength at which it really absorbs light really, really well. So we call this the lambda max for that particular molecule. So in the case of Coomassie Brilliant Blue, you'll recall that its lambda max was 595 nanometers. So if we back up here, you can see that, yes, Coomassie Brilliant Blue does have that maximum lambda value at 595. So for this reason, when we did the Bradford assay, we set the spectrophotometer to measure only at 595 nanometers, not only because that is the wavelength at which the Coomassie absorbs light the best, but because it's likelier that we're going to avoid hitting any absorbance values for other molecules that might be in the solution with the Coomassie. And then if you look at the picture here, you'll notice that for two different samples here, they both have the same lambda max, meaning that we're likely looking at the same molecule here, but we get a higher absorbance value for the more concentrated solution. The 5 milligram per milliliter solution only gets us an absorbance of 1, and the 10 milligram per milliliter solution gets us twice the absorbance at 2. So as you are, I'm sure, already aware, the general idea we want to work with here is that an increase in absorbance of a particular sample should indicate that the concentration has gone up in comparison. So for this reason, this is why when we did the Bradford assay, getting higher absorbance values indicated that we had more protein in the solution, both in the standards that we use to make our standard curve and also in the unknown bacterial lysates. Okay, so now let's talk a little bit more spectrophotometry theory. So this is going to be a little bit more involved. So at this point, you're already familiar with the idea that we are passing light through a sample that is loaded in a cuvette, and only a certain fraction of that light is going to make it through. What doesn't make it through gets absorbed by the solution. So if we're considering a very simple solution that only contains one type of solute, we can really use what is called the Beer-Lambert law, which is going to allow us to use spectrophotometry to directly measure the concentration of that particular solute. So the first thing that we need to kind of circle back to is the cuvette itself. I mentioned that cuvettes have basically an area at the bottom that is 1 centimeter by 1 centimeter. This is important because we want these cuvettes to be standardized so that light interacts with the solution over a known distance, which is usually going to be 1 centimeter for most cuvettes. So as light passes through the cuvette, it will be interacting with solute for exactly 1 centimeter of distance, and that is what we call the path length. Most molecules also have a particular characteristic called a molar extinction coefficient, which is given by the lowercase sigma Greek letter there. So this is a measure of how much light that substance will absorb per mole of solute. So like I said, this is generally speaking a unique figure for any particular solute. It's something that you can actually look up, which is actually very useful to us. So if we put all of this together and we look at this formula here, which is A, the absorbance, equals the molar extinction coefficient sigma times the concentration, which is what we're looking for here, and then times L, the path length, which we know is always 1 centimeter. If we get the absorbance of a particular solution, and if we know the path length, which we do because it's always 1 centimeter, and if we know the molar extinction coefficient, which we should because we can look that up, the only one of those 4 variables that we should not know is the concentration, and we can use very simple algebra to solve for the molar concentration in terms of moles per liter. This is really nice. Really, really nice. We don't have to do a standard curve. We can just straight up measure the absorbance of a sample. You have to blank the spectrophotometer first, of course, but we can just straight up get the absorbance of one sample and boom, simple calculation, we can solve for the concentration. However, this does have some limitations. The one that I mention here is that, unfortunately, this is only going to work for molecules whose concentration can be expressed with molarity. So that's probably going a little bit too far, but practically speaking, you wouldn't really want to use this unless you can actually measure something in terms of moles per liter, which is what molarity is. But the other issue with this, if you were asking, okay, so we just did the Bradford assay to measure protein concentration, we had to go through the whole rigmarole of making a standard curve, then we had to do all these calculations, we had to do all this nonsense in Microsoft Excel, plug our absorbances into the linear regression formula that we generated. It was a lot of hassle. So you're probably wondering, okay, why can't we just do this for protein? Why do we have to bother with the whole Bradford issue? Well, proteins can't really be expressed very easily in terms of moles per liter. And in addition to that, not every protein in solution is going to be identical to all the others. Whereas if you have, say, a solution of copper sulfate, which is something that appears blue, meaning that we can use spectrophotometry on it, all the molecules of all the copper sulfate in solution is going to be the same, so we can treat them all the same. But if you've got a mixture of a bunch of different proteins, not every protein is exactly the same. And we'll have more to say about that a little bit later on. But basically, there are limitations for using the Beer-Lambert law to directly measure concentration. So it's going to be a case of kind of knowing when you can and cannot do it. So let's look at a particular example here. So let's apply ourselves and say that we are making up a solution of a chemical called NADH. So this NADH molecule absorbs light at 340 nanometers. But unfortunately, when the scenario here is that when you were making the solution, you forgot to record the mass of the solute you weighed out when you made the solution. Oops. Yeah, that's kind of a big mistake to make. So if you don't know the mass of the solute, you can't possibly know the concentration. And you really have to know the concentration of a solution if you're going to be doing stuff with it. So you've got two options. You can either throw out the very expensive chemical and start over, which isn't really a great idea, or what you can do instead is you can use spectrophotometry to determine the concentration of the solution. So what we're going to do here is we're going to take this solution that you made, we're going to put it in a cuvette, and we are going to get an absorbance reading at 340 nanometers. So let's say that we blank the spectrophotometer first, and then let's say that the absorbance value that we get is 0.750. Okay, so now what we're going to do is we're going to use this formula for the Beer-Lambert law. We already know the absorbance, we already know the path length, which has to be 1 centimeter, and now we need to look up the extinction coefficient for NADH, which just so happens to be 6.22 millimolar centimeters inverse. And like I said, we already know the path length is 1 centimeter, so we know the absorbance, we know the extinction coefficient, and we know the path length. All we have to do now is solve for the concentration using this formula. So if we plug everything in, the absorbance is 0.750, the extinction coefficient is 6.22, concentration will leave as is because we don't know it yet, and the path length is 1. Well, if we solve for this, we get a concentration of 0.12 millimolar. So even though you screwed up a little bit and you forgot to record and remember what massive solute you were dealing with, by using spectrophotometry here, we were able to actually determine what the concentration of the unknown solution was. And that's a very powerful thing indeed. All right, so a couple more practical issues here. For NADH, we said its lambda max is 340. For Coomassie, we said it's 595. How do you go about figuring out what the lambda max is for a particular molecule if you can't just look it up? So the lambda max value for any given light-absorbing molecule, it's going to be a characteristic, and it's going to be worth knowing about. So if you don't know the lambda max, most good spectrophotometers, especially the ones that are computerized and are hooked up to computers, they can actually scan the molecule, meaning it bombards it with individual wavelengths all between the UV and the visible light spectrum, and it will graph out the absorbance at those particular wavelengths. So you can see we're scanning through from about 350 all the way up to 700, and for this particular molecule, the highest absorbance is given at this particular wavelength here. It looks like at about 450 nanometers. So all you have to do is just scan your sample at all the different pertinent wavelengths, and then you just point out what wavelength did I get the highest absorbance value, and that wavelength is going to be your lambda max, and that's going to be the wavelength that you'll want to work with in the future for this particular molecule. So the one lingering question is, do we always have to measure at the lambda max for a particular molecule? Well, the answer is a little bit complicated, as we're going to see on the next few slides. Typically, it is preferred to measure at the lambda max, but there will be some circumstances in which you may want to measure the absorbance at some other wavelength. So usually this isn't going to be the case, but sometimes it is unavoidable. So let's look at this particular case study here. So we're talking about two different very related molecules here, NAD plus and NADH. They are the same molecule, one is oxidized and one is reduced. So these are what are called cofactors. We talked a little bit about cofactors, I think, in, what was it, Chapter 5? So cofactors are chemicals that interact with enzymes and help to give them activity. So, for example, this enzyme here called alcohol dehydrogenase, its substrate is ethanol, which is grain alcohol, and the enzyme will actually convert that alcohol into acetaldehyde. So in order to catalyze this reaction, alcohol dehydrogenase, the enzyme, needs the cofactor NAD+. So as it catalyzes the reaction, NAD+ is reduced to NADH. So not only do we produce acetaldehyde in the process, we also produce NADH. So we can use spectrophotometry to monitor the progress of this reaction. Now, it has nothing to do with ethanol or acetaldehyde. Those are clear and colorless, and they therefore do not absorb light, so we can't use spectrophotometry to look at things that don't absorb light. But lucky for us, NADH and NAD plus do absorb UV light. So if you look at the absorption spectrum for both NAD plus and NADH, you'll notice that we get a lambda max at about 260 nanometers. So that might be your first instinct here. You might say, okay, we've got a lambda max at 260 nanometers, so we can study this reaction by reading absorbances at this particular lambda max wavelength, which is 260 nanometers. Well, do you see the problem here? Well, the problem is that both NAD plus and NADH have the same lambda max. It's right at 260 nanometers. So if NAD plus gets converted into NADH, then if they both absorb exactly the same at 260, then we're not going to be able to monitor the progression of the reaction because the absorbance is always going to stay the same, regardless of whether it's NAD plus or NADH. The absorbance should always be the same because the total concentration of both NAD plus and NADH should never change. So the conclusion we can make here is that measuring at 260 nanometers will not allow us to distinguish between NAD plus and NADH because they both absorb at that wavelength. So how do we get around this? Well, we're going to get around this by picking a different wavelength, one that is not the lambda max value. Look at the spectrum for NADH, which is in the dotted line there. So NADH, in addition to having its lambda max at 260, it also has a smaller peak at 340 nanometers. It doesn't quite absorb light as well at 340, but it does absorb a little. But you'll notice NAD plus does not absorb any light at 340. So if we instead pick 340 nanometers instead of 260, we can distinguish between NADH and NAD+, because if there's any NADH in the solution, we'll pick that up at 340 nanometers. And if there is none, then we should not get any absorbance readings. So doing things like this with enzymatic reactions is what we call a kinetic assay. We are using spectrophotometry to measure the production of either light-sensitive products or cofactors, in this case like NAD plus and NADH, and we can, as long as we're measuring changes in the absorbance over time, we can measure and look at and observe the progression of the reaction over time. So let's say that we put together in a test tube the enzyme, which is alcohol dehydrogenase, whatever buffer it's going to like to be in, ethanol, which is the substrate that it's going to be working on and turning into acetaldehyde, and then let's also give it this cofactor that it needs, which is NAD+. Well, at the start of the reaction, if 100% of the NAD plus is still NAD plus and we have not actually gotten any enzymatic reactions happening yet, the absorbance value should be 0 because we should not have generated any NADH yet. And keep in mind, we're absorbing at 340 nanometers. NAD plus does not absorb at 340, NADH does. However, as the reaction progresses, you'll notice that the absorbance starts to go up and up and up, and this is evidence of the enzyme not only converting ethanol to acetaldehyde, but specifically it's evidence that NAD plus is being converted into NADH, which does absorb at 340 nanometers. So as long as this reaction is continuing to go on, the absorbance value should continue to go up as the concentration of NADH in solution goes up as well. So there is one interesting thing here. You'll notice that eventually the absorbance kind of hits a maximal value here. It just does not go any higher than that. So what are some ideas that you could come up with as to why the absorbance stopped going up and plateaued like this? So you may want to pause the video here because I will be flashing some possible answers up on the screen in just a second, but you ought to be thinking about what is necessary for the reaction to proceed and why the reaction may have stopped like this. Okay, so the two probably most likely possibilities here is that whatever NAD plus that we started with, we probably got to the point where 100% of the NAD plus was converted to NADH. So it is not possible for us to make any further NADH So our reaction plateaued like this. The other possibility is that all of the ethanol in solution was converted to acetaldehyde, so no more substrate, well, no more reaction either. So both are equally likely, but just given the limited amount of information here, we can't really distinguish between the two. So the last thing that we want to kind of talk about here specifically in this case is limits to spectrophotometry. Spectrophotometry is great, but there are limitations that we need to talk about. So just like what we saw with the Bradford assay, we get the idea that the more concentrated a sample is, the more light is going to be absorbed. But there is a limit to how much light can be absorbed. So the sample will reach its max absorbance when you get close to 100% of light getting shown through the sample being absorbed. Based on the equation that we looked at earlier, the max theoretical absorbance is 2. So no matter how concentrated a sample is, if the absorbance value gets close to 2, you're not going to be able to distinguish between this sample and an even more concentrated sample, because at a certain point, the solution is so concentrated that no light can pass through it, which gives you your max absorbance. So generally speaking, we really don't want absorbance values getting much higher than 1.00. Once the absorbance gets much past 1, the relationship between absorbance and concentration stops becoming linear. We want, for our practical calculation purposes, we want that relationship to be a straight line like what you see right here. If the absorbance values get too high, then that relationship stops being linear, and our approximation of the concentration is going to become much, much, much less accurate. So, practically speaking, if you do some spectrophotometry and you get an absorbance value that is greater than 1, you should not be confident in your ability to get an accurate concentration from that sample. So what do you do with that? Well, what you would do is you would take your sample that you are trying to measure the concentration for, you just need to dilute it down. So if your first attempt gives you an absorbance of like 1.5, well, if you dilute it down tenfold, meaning you add 1 part of your sample to 9 parts water, then your next try should give you a much, much, much lower absorbance value, something that you can actually work with. So you can then calculate the concentration of that diluted sample and then reapply your dilution factor. So if you diluted it 1 to 10, you can just multiply your calculated concentration by 10 to then figure out the original concentration. So basically what you should walk away from this slide thinking is that if I get an absorbance greater than 1, there are more steps that I need to take. I can't just calculate the concentration straight from that. It's not going to be accurate. Okay, so we're going to go ahead and wrap up this video here. The last video for this particular chapter, we're going to talk specifically about measuring the concentration of proteins and DNA samples using spectrophotometry. So thanks for your attention for this unusually long video, and